So yes, so I'm a person, I study phytoplankton primarily in polar regions. And so, as Scott said, I started out in the Antarctic. I am now working in the Arctic. We use a variety of techniques in my research group. These are satellite-based studies. So these are the first ever large-scale estimates of productivity in both polar regions. And we don't just do satellite work, we also do field work, and we do a lot of modeling work. And so I'm just going to show you a couple quick studies that we're really excited about that we've done recently. The first one is this. And so what I'm going to show you is, under ice phytoplankton blooms. And so it's something that no one had ever heard of for a long time until we went on this particular expedition. And what you're going to see is this is a video produced. You're going to see it's going to zoom into the ship, and you're going to see how we sampled the bloom in real time. And so here we're coming into it. I hope you can see this. It's maybe a little washed out because of the color. There's the icebreaker. So we're in the open water now, and the white that you're seeing is the sea ice. So we're sampling in open water. What it's going to do, it's going to flip down, and hopefully you can see areas where we're actually doing the sampling, the depths that we're sampling at, and eventually what you're going to see is colors are going to appear. Purple is basically no phytoplankton at all, very low concentrations. Red is very high. Open water, there's very little phytoplankton. We are now headed toward the ice, and suddenly we're seeing very much higher concentrations of chlorophyll. These red values are extraordinarily high. This region right here, that spot, is the largest concentration of chlorophyll measured anywhere in the world, ever. And this is under a meter of ice. And it's something that we thought was not possible. We got very excited on seeing it on the way in, so we turned around and went back out again and resampled it again, only we sampled it at a much higher resolution, and we caught that bloom again. And so there it is. It's about 30 meters thick, and it's pea soup green. It's the greenest water I've ever seen in my life. We headed back out into open water again, and the concentrations start to drop again. So here, the action is happening under the ice. As I said earlier, we do satellite-based studies. We are completely missing this. We can't see them because it's under ice. Satellites don't know that they're there. And so our estimates of production for these waters are off by a factor of 10. So we need to figure out a way to sample these things. And that's one of the things we're working on. But we haven't figured it out yet, and I'm not going to talk about that today. So that's one thing we're doing. Down in south, what we're working on, this is a cruise we're going to be taking we're going in December. And so what this is a satellite image of phytoplankton again. Red is high concentrations. And there's this big phytoplankton bloom, and that's where that map is. You can see right there, right off the coast of Antarctica. And there's this big phytoplankton bloom. It's the middle of the Antarctic circumpolar current in a place where you wouldn't expect to see a big phytoplankton bloom. There's really no other high chlorophyll concentrations anywhere around it. Farther north there is, yes. But it turns out this phytoplankton are located right above two hydrothermal vents. And when we sampled that, we found out that there was, and we sampled it in 2014, that there's really high concentrations of iron right under those blooms. So what it looks like is happening is iron from the hydrothermal vents is fertilizing the ocean and causing increased phytoplankton concentrations. And so we're going to study this in detail in December because all this evidence is circumstantial, and we'll see if that's really what's happening. But one of the things I'm really interested in is, all right, primary production. Phytoplankton are producing all this organic carbon. What actually is happening to this organic carbon? Well, as an ecologist, I'm interested in the fact that that feeds the entire food web. All the marine food webs ultimately rely on the carbon produced by phytoplankton. So that's important. But what they also do is they are the main contributors to what we call the biological pump. And the biological pump refers to the fact that phytoplankton in the surface ocean take up CO2 when they photosynthesize. Eventually they die and they sink and they take that carbon with them, not all of it, but some of it. And what that does is it creates a CO2 deficit in the surface ocean that is then replaced by atmospheric CO2. So they are essentially conduits for carbon from the surface ocean down to the deep. And so it's really important ecologically, but it's also receiving a lot more attention recently in terms of a potential geoengineering solution for climate change. In that if we can artificially stimulate the production of these phytoplankton, maybe we can use that as a way to get CO2 out of the atmosphere. Now this sort of had a heyday 20 years ago, and we kind of forgot about it and ignored it. It's come roaring back now because our problems have gotten so severe that even things that seemed harebrained back then are becoming more potentially possible now. And so looking at the potential for this is really important. The question is, can this work? And so there's lots of ways to do this. People are proposing a lot of things. The most logical thing to do is to fertilize some areas of the ocean with iron. So this is a map of surface nutrient concentration, in this case nitrate. And there are places in the world where there's plenty of nitrate fertilizer for phytoplankton, but there's not enough iron. If you add iron to these places, phytoplankton will grow. And so the Southern Ocean is one, the Equatorial Pacific is another, the North Atlantic is another. Do back of the envelope calculations. Let's just assume that we add enough iron to these waters to remove all that extra nitrate. How much carbon can you sequester? And for the Southern Ocean, it's about 0.5 petagram carbon per year, which is a decent amount. I know a lot of the solutions would like to see that be a teragram, or 1 petagram, but it's not. And then these other areas are a little less. So that's just a back of the envelope calculation. Lots of issues with this. One of them is cost. How much is something like this gonna cost? Because you basically have to take tanker ships and spread iron all over the Southern Ocean every single year. And so you don't need to go through all these. These are just some of the back of the envelope costs. If you count how much does it cost to operate a ship, you need 260 ships. You're gonna need to do this for a month every year. You need to buy the ships, you need to buy the iron. You put all those together and it comes out to be about $6 a ton of CO2, which when you compare that to other geoengineering type solutions, looks pretty good. It's not bad. Now, $6 is an absolute minimum estimate. It doesn't include the cost of validation, verification, all the other things that go into it, and that's where that $6 to $100 range comes from. But it makes it an attractive option. The thing I always worried about was, all right, we got to spread this stuff out with ships. Those ships are burning diesel fuel and they're releasing carbon. Are we actually going to end up burning more carbon than we sequester by doing this? And so did the math, and it turns out it's only going to burn 0.013 petagrams. So it's completely insignificant. And so this was what I thought was the biggest problem with this solution, and it turns out it's not a problem with this solution at all. And so it's not that expensive, and it's potentially not that harmful in terms of increasing CO2 emissions, because if you're going to sequester 0.6 petagrams and it only costs you 0.013, it's not a bad trade-off. But the big question is, yes, we can increase phytoplankton production, but do you get export? And that's been the biggest problem with this. Will those phytoplankton export extra carbon to the deep ocean? These experiments have been done a dozen or so times around the world, and there's been only one experiment that has shown unequivocally that there's actually export of carbon, that you're actually sinking carbon to the deep sea. Every other experiment, that carbon that was produced just got outgassed back to the atmosphere. Now, most of these cruises were just not there for very long, and so maybe if you could sit there long enough, maybe you could tell. So what controls how much export there is? Well, it's how fast these things are sinking, right? And so that is the number one factor. How fast do they sink? Big cells like this sink really quickly. Little tiny cells like this sink very slowly. Obviously, if you fertilize the ocean, you want to grow these cells. You don't want to grow the little ones there. But there's another part to this, and that is, it's just not how big they are that determines whether they sink or not. Phytoplankton can regulate their buoyancy. They can determine themselves whether they sink or they rise. And so this is an example of a diatom that sinks down into the deep ocean to get nutrients, and it rises back up to get light, and it sinks to get nutrients. And so phytoplankton can do a lot more things than we give them credit for. So we studied this recently in the Arctic. We went back to that under ice phytoplankton bloom, hoped that we would find one, and we did. And so we did this project Tale of Three Systems we call TOTS. And the three systems are, we're looking at ice algae, we're looking at under ice blooms, and we're looking at spring blooms. And we want to see, how do these things, how do they compare in terms of their ability to regulate their buoyancy, and how fast do they sink through the water column? And so this is where we went. So we started in Nome, we went up, and we essentially, the crew thought we were crazy, because we just did this trapezoid 7 times, around and around and around and around. But that was great because we got this long time series from the same location. And so what we had on the ship, this was done with Manu Prakash. This is his gravity machine. I don't know if you've all seen the gravity machine before. Probably you have. But what it allows you to do is you can measure the sinking speed of an individual particle as it sinks through this infinitely long wheel of water, right? And you can focus on it. And so this is going to be a video of one there. Let's see. Oh, there we go. So there's a diatom. You can see it's sinking through the water column. And then it just abruptly stops. So these things can regulate their buoyancy on millisecond time scales. It's going to sit there for a minute, and then it's going to sink again, right? And it just does this over and over again. This particular one wasn't rising, but they can do the same thing. They can go up, they can stop. So these things are really good at regulating their buoyancy. Now, we've got 7,000 videos that we've got to analyze, and we're just in the process of doing it now, so these results are still early. But the one thing we notice is that sea ice algae are little rocks. They sink really fast, right? They have very little ability to regulate their buoyancy. There were no positively buoyant cells from the sea ice. But once we got to the under ice bloom, they sank much slower, and some of them were positively buoyant. In the open ocean, a lot more were positively buoyant. It's that moving up and down to get nutrients and light when you're in a deep ocean. So some sink fast, some sink slow. The other thing we discovered, which was a big surprise, is that there's physical mechanisms that actually pull this phytoplankton bloom from the surface ocean all the way to the bottom. So these lines are lines of constant density, and that's what water flows along, lines of constant density. And you could see that the yellow color there, which is high concentrations of chlorophyll, being pulled all the way to the bottom. Well, we measured sediment chlorophyll concentrations, and you can see that all this chlorophyll is accumulating in the sediments. And so it's not just biology, it's also physical mechanisms that are resulting in increased export of carbon from the surface. So the consequences for the Arctic are huge in terms of whether or not these things sink fast or slow. If they sink slowly, that gives the zooplankton lots of time to eat them, which are eaten by fish, which are eaten by bowhead whales that the indigenous populations use for their food. If instead these cells are sinking very quickly, those things reach the bottom, where they feed clams and amphipods and other things, which are fed upon by things like gray whales and walruses. So it's a completely different ecosystem when these cells are sinking fast versus when the cells are sinking slowly. So what we're going to do, what our plan now is to take this information and put it in our model. We've got a model, full ocean ecosystem model that includes the sediments, and start incorporating sinking behavior into these models. So we can do a better job of both predicting ecosystem function, how likely are we to get a switch from one ecosystem type to another, but more importantly, we can use it, or maybe not more importantly, but as importantly, is it can be used as a tool to start evaluating the efficiency of export potential under different situations, because we need to be able to incorporate this kind of behavior if we're going to get the right answers in terms of whether these geoengineering solutions will work or not. And I will stop there and be happy to answer any questions.